Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to the channel Anatomy Canvas. Today in Embryology Foundation module we are going to discuss with the process of the gastrulation. So third week of development is characterized by three features, that is appearance of the primitive streak, the development of notochord and differentiation of the three germ layers. First, have a look on this diagram, and this is a little bit revision, that is conceptus with the bilaminar embryonic disc. So in this diagram you can see there are three main cavities are present. One is this large cavity called the chorionic cavity, and this cavity is surrounded by the extraembryonic mesoblast cells, then the cytotrophoblast and syncytiotrophoblast, and this cavity is surrounding the two small cavities inside the embryonic disc. These are the amniotic cavity and the definitive yolk sac. Another fourth cavity, the allantois, will form as a hypoblastic diverticulum at day 16, and this you should remember that this is also your MCQ as well. Also little bit revision over here about the syncytio and the cytotrophoblast. So you can see this is the lacunar stage in which lacunae are forming in the syncytiotrophoblast, and primary villi are formed, which are formed by the cytotrophoblast, which are covered by the syncytiotrophoblast. So what is the gastrulation and how the primitive streak is formed, which is an important feature in third week of development. Early development of the period of development, during which significant rearrangement, migration and folding of the early embryo occurs. So in third week of development, at the start there are certain features with that is the significant rearrangement of the cell, migration of the cell, and also little bit folding of the early embryo. So what is the gastrulation actually? That is the process that establishes all the three germ layers. Up till now we have studied that the embryonic disc is bilaminar and it consists of the two germ layers. The one is called epiblast, which is facing the amniotic cavity, and other is the hypoblast, which is forming the roof of the definitive yolk sac or umbilical vesicle. So during the process of the gastrulation, the main important feature is the appearance of the primitive streak. So primitive streak appears as an opacity formed by thickened linear band of the epiblast. So this is an opacity in the thickened linear band of the epiblast and as a collection of the pluripotent cell, and this is oriented along the long axis caudally in the median plane on the rostral aspect, on the dorsal aspect of the embryo. So this layer is actually epiblast, and we are looking at the surface of the epiblast. We have removed from here the amniotic cavities. These are the edges of the amniotic cavity, and we are looking at the epiblast. In the epiblast, on its dorsal aspect, on its caudal direction, in the median plane there is an opacity which is formed by the proliferation of the pluripotent cells. So this is the primitive streak, and this streak extends halfway along the disc, reaching its greatest relative length at pre-somatic stage, that is stage 7, and maximum length at the stage 13. So these are the cells which are migrating, these are the pluripotent cells which are migrating through the area of the primitive streak, and they come to lie in between the epiblast and hypoblast layers. So gastrulation, the proliferation and migration of the epiblast. As the epiblast cells proliferate, the two ridges are formed, and in between two ridges there is a groove, which is the indentation in between these two ridges, and this groove is called the primitive groove, which is present in the primitive streak. The most cranial portion, the most rostral portion of this primitive streak is called primitive node, and indentation indentation in this primitive node is called the primitive streak. So these are the few definitions which you must understand, that this is the primitive streak. The indentation in the primitive streak is the primitive groove. This elevated area is primitive node, also called the Hansen's node, and depression in the primitive node is called the primitive pit. Another diagram to understand this is we are looking at the surface of the epiblast on its dorsal aspect of the embryo. So this is the area of the primitive streak, and in the center of the primitive streak you can see the primitive groove is present. This cranial part of primitive streak is primitive node, and in the center of the primitive node you can see primitive pit. This area is the precaudal plate, and this area will form later on the embryonic endoderm. Here you can see the cut edge of the amnion as well. The section taken at this level shows that there is indentation in the primitive streak, which is called primitive groove, and cells are migrating from the primitive streak. These are the cells are derived from the epiblast cells. They show epithelial to mesenchymal transition, and they come to lie either in between the epiblast and hypoblast, or they even displace the hypoblast and comes to lie over here. So another diagram in which you can see that when the cells displace the hypoblast cell over here, initially they are intermingled between them, and at this stage you can see either endodermal cells and notochordal cells are present, which are the cells derived from the epiblast. This area is the intermediate area which will form the mesenchyme. Later on it will form the mesoderm, and this epiblast will later on form the ectoderm. So by the process of the gastrulation, the epiblast will form all the three germ layers: the ectoderm, the mesoderm, and the endoderm. In this diagram you can see this is the area where the migrating cells are present, and these migrating cells are pluripotent, and this is epiblast is actually the cells are epithelial in nature, and they show a transition from epithelial to mesenchymal, and then they migrate and come to lie between epiblast and hypoblast. So what is the process of ingression, also called invagination? So the process by which cells become part of the streak and then they migrate away from it beneath the epiblast. This is termed as ingression or invagination. So the process by which the cells are migrating and they slip beneath the primitive streak and come to lie beneath the epiblast. So the process is called either ingression or invagination. These cells become flask shaped and slip beneath the epiblast. So important factors which are involved over here, which are responsible for epithelial to mesenchymal transition of these epiblast cells are: number one, fibroblast growth factor 8, and this fibroblast growth factor 8 down regulate the protein E-cadherin protein. This E-cadherin protein is responsible for the junctions between the epiblast cells, and epiblast cells are present as the ectodermal cells. When there is a down regulation of the E-cadherin, these cells loosely arranged attached to each other, then they become flask shape, and then they starts to migrate. So passage through the streak is specified according to position. This is also very important. The cells which are migrating via node or cranial or middle or caudal regions of the streak, which will form the specific areas. So it is also called the fate map of the primitive streak. Here you can see this is the area of the primitive node, and the cranial most cells which are migrating through the primitive node will form the notochord and the endoderm. The cells migrating through this area of the node will form the medial half of the somite, or you can say will form the praxial mesoderm, its medial part, and cells migrating through the cranial part of the primitive streak will form the lateral half of the somite. You can also say the lateral part of the praxial mesoderm. Cell migrating through mid portion will form the mesoblast or the intermediate mesoderm, and the cells located over here will form the primordial germ cells, and the caudal most part will form the extra embryonic mesoblast or extra embryonic mesoderm. So this is called the fate map of the primitive streak. The cells which do not migrate through the epiblast, through the primitive streak, and then they remain in the epiblast, they will form the neural ectoderm and the surface ectoderm. So these are the different cell population which migrate through either the primitive node or the primitive streak, and their passage through streak is specified according to their position. So another structure very important is precaudal plate. What is the precaudal plate? It is defined as localized thickening of endoderm rostral to the notocaudal process. In this diagram, you can see this is the notocaudal process, and this area is called the precaudal plate. This is the localized thickening of the endoderm cranial to the notocaudal process. Although it is seen as a highly developed mesenchymal mass and it is in contact with the floor of the neural groove, you can see this is the area of the neural groove, and this precaudal plate is in contact with the floor of the neural groove, which is cranial to the notocaudal process. In stage 8 embryo, that is nearly day 23, the precaudal plate is up to 8 cell deep and extends along the long axis of the embryonic disc, but its height decreases later on, and at stage 9 to 10 the lateral edges of the plate have begun to migrate laterally as free mesenchymal cells, and plate reduces in height to 2 cell deep. So at the stage 8 embryo, that nearly 23rd day of development, the precaudal plate is 8 cell deep, and stage 9 to 10 it is only 2 cell deep. It is responsible to give rise to endodermal layer of the buccopharyngeal membrane later on. So you can see this is the precaudal plate. This is the floor of the neural groove. Here you can see the intraembryonic coelom which is forming. Here you can see the notocaudal plate overlying ectoderm. This area is the neuroenteric canal, which we'll study in a while, and this area is the primitive streak, and here you can see the endoderm which is forming and the cloacal membrane which is also forming over here. This is the diverticulum which is called the allantois, and this allantois is formed at day 16. Another important point to remember over here is that this allantois will later on form the urachus, and in the newborn babies this will be represented as the median umbilical ligament. So all the points related to allantois are your MCQs. Please remember that it appears on day 16, your first MCQ. It will form the urachus, second MCQ, and it will form the median umbilical ligament, your third MCQ. So this is the diagram. Here you can see the bilaminar germ disc is present, which is consisting of the epiblast and hypoblast. The epiblast is facing the amniotic cavity and hypoblast is forming the roof of the umbilical vesicle. Here you can see the connecting stalk. In this diagram also you can see the primitive streak now has started to appear, and in this section you can also see the primitive streak, and this level of the section D here you can see the cells starts to migrate and slip beneath the epiblast, and they come to lie between epiblast and hypoblast. So here you can see in this diagram the primitive node is present, primitive streak and primitive groove is present, and cells are migrating. They are slipping beneath epiblast and they come to lie on either side, cranial as well as caudal direction. Important clinical correlate over here is the sacrococcygeal teratoma. So you must remember that after the formation of the 3 germ layers, when the process of the gastrulation is complete, at the end of the 4th week, the primitive streak usually disappears. If it do not disappear and it remains itself, it can lead to anomaly called the sacrococcygeal teratoma. So you can see over here, this is the sacrococcygeal teratoma. So teratoma are the type of the tumors which contains all the three germ layers, and you people know by the process of the gastrulation, as epiblast is responsible for formation of all the three germ layers, and the remnant of the primitive streak will be able to form all the tissues derived from the three germ layers. So this is called the sacrococcygeal teratoma. Now we move on to the process, the notochordal process and notochord formation, which is an important event also in the third week of development. So in this diagram, first you can see these are the this is the notochordal process, the cells migrating through the primitive streak, and they come to lie between the epiblast and hypoblast up to the level of the precaudal plate, and these cells are forming initially the notochordal process. So you must remember, first of all, notochordal process is formed. Later on, the canal of the primitive pit also enters into the notochordal process, and then notochordal canal is formed. So you can see after the notochordal process, notochordal canal will form. This is the section taken at this level, and in this section you can see the notochordal canal is present, and on each side of the notochordal canal you can see the intraembryonic mesoderm is present. This notochordal canal extends up to the level where the notochordal process was present. It was formed till the precaudal plate. So what happens next? That when it extends over here, the lower layer of the notochordal canal then intermingle with the embryonic endoderm, and when these two layers fuses, this layer degenerates. As a result of degeneration, temporary connection is formed between the amniotic cavity and the definitive yolk sac. This temporary connection is called neuroenteric canal. Here it is written, this is the neuroenteric canal, and the process now formed is called the notochordal plate. So you should remember, first notochordal process is formed. After notochordal process, the notochordal canal will form. After the disintegration of the lower layer of the notochordal canal, only the thing which remains is the notochordal plate. What happens next? That this notochordal plate later on starts to invaginate, and it forms a rounded tubular structure along the long axis of the embryonic disc, and it detaches from the endoderm, and endoderm over here is complete again, and this notochord is complete, and it is called now the notochord. So in this diagram you can also see here the neural groove and neural plate is formed. You people, you students always confuse the process of neurulation along with the process of the notochord formation. So please remember, notochord, also called carda mesoderm, also it is formed from the epiblast cells which are migrating through the primitive streak, while the neural tube is formed by the cells which remains in the epiblast, which do not migrate through the primitive streak, and which are formed by the inductive influence of the notochord, and this layer of the ectoderm will form neuroectoderm, and later on it will form the neural plate. Here another beautiful diagram you can see. This is the area where temporary neuroenteric canal is present. Here you can see the notochordal process is forming. Here you can see the precaudal plate is present, the mesenchyme, the ectoderm is forming, the primitive streak, intermediate mesoderm over here, and layer of the hypoblast which will later be replaced by the embryonic endoderm. And this is the hypoblastic diverticulum called the allantois. Another important point in the third week of development is the caudal eminence. So, students, you are always confused when you study the caudal eminence in your book. So, you are confused what is the caudal eminence actually is. So, the caudal eminence is actually the region between the neuroenteric canal and the cloacal membrane. So this region from the neuroenteric canal and the cloacal membrane, including the primitive streak, and this area is called the caudal eminence. It consists of caudal region of the trunk composed of mesenchyme derived from the primitive streak. So this is the mesenchyme also, which is derived from the primitive streak, and the epiblast covered with the surface ectoderm. This was initially the primitive streak, which will later form the surface ectoderm. Cells of the caudal eminence arises from localized division of mesenchymal population positioned caudal to the neuroenteric canal. So this area is the caudal eminence from the neuroenteric canal to the cloacal membrane. So to revise the whole today's lecture, the events which are occurring in the third week of development. So first was the formation of the primitive streak, then the formation of the notochord, and then this primitive streak of by the process of the gastrulation, the epiblast cells are forming all the three germ layers. So you can see this is the surface ectoderm which is forming, which will later on also form the neural tube. Here you can see the neural groove as well. The cells which are present over here will form the buccopharyngeal membrane derived from the precaudal plate. These are the notochordal plate, which are also called the cauda, mesoderm, which are derived from the cells migrating through the primitive streak, and also other cells which are migrating through the primitive streak, which beneath on each side of this primitive neural plate will form the intermediate mesoderm, which will form the mesoderm, the second layer over here, and the cells which are going to replace the hypoblastic layer will form the endoderm. So by the process of the gastrulation, the epiblast will form all the three germ layers, the ectoderm, the mesoderm, and endoderm. An important event in the third week of development are first the primitive streak, formation of the primitive streak, then the formation of the notochord, the precaudal plate, and formation of all the three germ layers. Here I complete my today's class, and this was a little bit difficult lecture, and you must revise it to memorize it. So here are the references, that is Langman's Medical Embryology, The Developing Human by the Keith L. Moore, and The Gray's Anatomy by Susan Stranding, the 40th edition. So I must say you should study this topic both from Langman as well as from The Developing Human by the Keith L. Moore to have a clear concept. Thank you very much for your attention, dear students, and inshallah we will meet in the next class. Inshallah after that we will do the neural tube formation. Till then, Allah Hafiz.